So with that, I'd like to introduce our next speaker, who's going to be Bill Noble. And I also wanted to ask if you have an empty seat next to you and are willing to kind of move down and make room for some people on the edges so more people can file in, that would be awesome. But so Bill Noble is joining us here from UW today, which is great.
All right, thank you very much. So my background's computational, kind of similar to David, but I'm going to be talking about DNA rather than proteins. Most of the work I'm talking about today is done in the context of the UW Center for Nuclear Organization and Function, which is a center that Jay Shenduri and I lead, along with Chuck Murray, who's here, Christine Desteche, I'm not sure if she's here, and some other investigators. So this is part of the NIH 4D Nucleome Consortium, and the consortium involves labs doing sequencing assays and imaging assays to try to understand the three-dimensional structure of the nucleus over time. So what I'm going to focus on today are two particular projects. The first one is done by a postdoc in my lab, Gurkan Yardimchi, along with Jijun Duan, Jay Shenduri, and some people in hematology and lab medicine. We're looking at how the three-dimensional structure of DNA changes during blood cell differentiation. So I'm sure most of you are familiar with the different stages of hematopoiesis. We're showing here just the red blood cell and the white blood cell lineage. What we've been doing is assaying the three-dimensional architecture at 3 different time points during each of these lineages so that we can try to characterize how that architecture is changing through differentiation. So I'm sure most of you are familiar with the Hi-C assay, but just in case, so we're all on the same page, we're using sort of the workhorse of the center is this assay Hi-C that was developed back in 2009 was the first description of it. The basic idea is it's a cross-linking assay, so you're introducing formaldehyde cross-links. These days it's mostly done in situ. And then there's various cleaving and marking steps, but the end readout is you're using DNA paired end sequencing to identify pairs of DNA loci that are distal along the genome sequence, but are close in Euclidean distance within the three-dimensional space of the nucleus. So when you run one of these assays, you get tens of millions or potentially up to billions of paired end reads. And these are typically organized into a matrix. So the dimensions of the matrix correspond to the length of the genome. This is a yeast genome here. You can see there's a strong diagonal because you have lots of contacts within chromosomes. So each dot in the matrix, each entry in the matrix is a count, essentially of how frequently within this population of heterogeneous cells, these two DNA loci were close together, close enough to be cross-linked by the formaldehyde. So what we've been doing is looking at various properties of the DNA architecture over time. And as in many fields, one of the things that we have to do is sort of simplify the complex structure into sort of concepts that make sense to the human mind. So one organizational structure that we look at is something that's called a compartment. This was actually first described in this 2009 paper, and the concept is that if you look at the way the DNA is organized, there are these 2 categories of regions of the genome. They're multi-megabase regions. They're called A and B, or active and inactive compartments. And the key observation is that if you look at the 3-dimensional contact patterns, things that are in the A compartment tend to interact with other things in the A compartment and vice versa. And so our question is, what's going on with these compartments during the blood cell differentiation? So we're just looking at overall correlation between or actually this is just percentage of shared compartment structure between cells in the different stages. So we have the 3 stages of the white cell lineage here, 3 stages of the red cell lineage, and not unexpectedly, you get the strongest correlations within lineage and less similar compartment structures between lineages. But the question is, what does this mean functionally for the cell? One of the key characteristics of compartments in the 3D structure is that the active compartment is strongly enriched for genes, for enhancers, and so on. We see that very clearly here. So this is for each of these different stages, the active and the inactive. We took a set of annotated enhancers, and you look at the enrichment, it's very strong for the active enhancer for the locations of the enhancers. But what you may also notice is that there's a downward trend within each of the lineages here, and essentially an upward trend within the active part. So the compartments are, both the compartments are changing, the compartments are changing during differentiation to increase the density of active enhancers within those regions. We see a similar kind of pattern with histone modifications. If you're familiar with all these different names of the histone modifications, essentially what you can see is that we have a fairly consistent enrichment of activating histone marks in the active compartments, with the exception of the H3K9ME3, which is, of course, the opposite way around. And you see this both in the red blood cell and the white cell lineage. One of the things we're particularly interested in is looking at particular loci and seeing if what's happening at those more focused loci agrees with what we knew a priori. So for example, if you look at GATA1, this is obviously a key gene for the red blood cell lineage, and what you can see is that it's active in the, it's inactive, sorry, it's in the white blood cell lineage. It's inactive in the red blood cell stages and then becoming active later on in the white blood cell lineage. We also looked at several other loci, for example, beta globin locus, which is active in red and inactive in white, and the AQP9. The compartment scale, however, is only sort of the largest scale that we look at in the chromatin architecture. And one of the features that people have been looking at recently is something that's been termed a FIRE, or a frequently interacting region. The name is exactly like what you'd expect. If you zoom in on a small part of the high C contact matrix, you can essentially look for a DNA locus that shows an enrichment of contacts with lots of other places along the genome. So it's drawn here as this cross. You don't really focus on the nearby contacts. You're looking at sort of more distal contacts and finding regions where you see a lot of those contacts. So we can systematically identify these FIREs at all of the different stages. And the interesting thing is that if you just take the FIREs, pull out the genes that are in them, and run them through one of these gene ontology tools like GRATE, you'll see immediately that the terms that pop out are exactly the kind of things that you'd expect. Heme pathway and various erythrocytic terms for the red blood cells. For the white blood cells, you see things like inflammatory response, adaptive immune response, leukocyte activation, and so on. We can also see that if we look at the actual gene expression within these FIREs, you see a very strong enrichment. So this is just log the false discovery rate of the fold change of the RNA-seq data compared with differentiated red blood cells. And there's strong enrichment in these three stages here. And if we compare the amount of enrichment with a bunch of other cell types, you don't see nearly as much enrichment, except, of course, in a couple of cell types that are similar. So what we're looking at now is moving on from this kind of bulk analysis, which is looking at a heterogeneous population of cells, to start using the single cell high C assay, which was a variant of which has been developed in the Shenduri lab. The single cell high C allows us to characterize not just what the population looks like, but changes in single cell architecture from one cell to the next. In particular, we're interested in trying to understand in the mature white blood cells how the chromosomes organize into different lobes in mature white blood cells, and we can look at that with the single cell technology. Now, that's been mostly still at the high level, looking at large-scale phenomena, but one of the things that we're interested, especially as we try to characterize the variance or the changing nature of the 3D architecture, is how can we characterize what's going on at specific loci in more detail. And for this, Dejeune, who's a postdoc jointly with me and Cole Trapnell in genome sciences, has been bringing to bear methods from biophysics, which is what his PhD is from, looking at how we can model an active chromatin hub. So in this case, we're actually, at this point, not using Hi-C data, but actually using a different kind of data that I'll tell you about in a second. So I'm sure most of you are familiar with this, but the beta globin locus is a sort of a classic example of the active chromatin hub model. So you have in the erythroid or early stages, you have these set of 6 hypersensitive sites, 3 of which are in the hub and 3 of which are exterior. You have these 4 relevant genes. And at different stages in the erythroid cell, whether you're in the primitive stage or the definitive stage, you get 2 of the, well, so all 6 of the hypersensitive sites in the hub, and then 2 of the genes get recruited, and then 2 other genes get recruited at different times. So the question is, can we use some data and computational modeling to better understand what's going on in this hub? So the kinds of questions that we're interested in asking are, how is gene positioning related to, in the hub related to gene activity? How's the order of structural change related to hub formation? And what about the set of events required for gene activation? So the data that we're actually using is a single cell data type, but it's not the Hi-C assay, it's a TAC-seq. So a TAC-seq is a method for measuring open chromatin, essentially hypersensitive sites. So what each of these rows represents is one cell, and the data here is each peak represents one hypersensitive site, so these are canonically around a couple hundred, 250 bases wide or so. And canonically what we have is this kind of data derived from thousands of different cells. And so you have the genomic position here, all this pattern of hypersensitive sites along the genome. And we're using a tool that was developed in Cole's lab called Cicero by Hannah Pleiner. And what Cicero does is looks at the pattern of co-activity between pairs of hypersensitive sites across the population of cells. Essentially, the concept is if in any given cell, this pair of hypersensitive sites is either on all the time or off all the time, but never in a one-on, one-off kind of state, then you can infer that there must be some kind of relationship between those, some kind of physical relationship that allows them to be in the same state all the time. It's a little more complicated than that because you have to tease out the transitive properties and identify the ones that are directly responsible for each other. But suffice it to say that the output of the Cicero model is a matrix like this, where for every pair of entries in the matrix, you're looking at a pair of hypersensitive sites in the genome, and you're essentially characterizing with a score the co-accessibility of those 2 sites. So the question is, can we use this matrix as input to some kind of model? The challenge is that if you look at what's going on, for instance, at this beta globin locus, here's the 2 stages that we're familiar with, and here's the Cicero model just showing where there's, we sort of binarized the scores here for simplicity, but what you can see is that it's kind of a mess in the sense that, it's not like the active chromatin hub model just falls out directly from this. And the question is, does that mean there's something wrong with our data, something wrong with the model, or what's going on? And this is where we turn to the polymer modeling. What Dejeune has done is used sort of standard molecular dynamics procedures to build a polymer model where each monomer represents sort of a nucleosome-sized piece of DNA, and then deriving the basic parameters of the model from previously reported experiments. And the key idea is that the Cicero contacts are modeled as these attractive springs, where the spring constant is proportional to the score that comes out of Cicero. So when we do that, we get a model like this. So you can see here, these are all the different, the hypersensitive sites here in white and purple, and then you have the different genes in the active chromatin hub. If you look, here's a model of the early stage, and if we set it running, qualitatively what you see is it's sort of all over the place, right? Things are moving in a fairly wild way, and in particular, the colored bits are not really all close together. If you contrast that with the later stage, you can now see that things are much more compact. There are, as the structure moves, you still see things grouped together. If you pull out one frame from each of those 2 movies and put them side by side, the key observation is that you have the active chromatin hub over here and things coming in and out of it, essentially what you see is that the fetal stage genes are not really excluded during the adult stage, but overall, on average, there is less they talk less in the adult stage than in the fetal stage. So it's obviously hard to stare at those pictures and see what's going on. So what we do is statistical analysis of the resulting movie. So this is just showing that you have the fetal stage genes here, the adult stage genes here, and we're looking at the distance from the 5' hypersensitive sites, right? And so in this setting, you can see there's a difference here. Essentially, the key observation is the white space here between these two. So in that setting, you have a separation, whereas if you look at the 3' hypersensitive sites, in contrast, you no longer see this separation between the two. And so the idea is that we can use this kind of molecular dynamics model in conjunction with primary data to answer questions about what is going on in a more detailed sense than sort of the relatively simplistic stories about things being outside in one stage and inside in another stage. So the current work that we're doing now is how do we incorporate other information? Clearly, the next stage will be to incorporate Hi-C data, but we also have access to lots of other epigenetic data characterizing things like histone modifications and so on. And so the nice thing about having these kinds of physically plausible models is that they provide you a structure and a framework for incorporating orthogonal data types into a single unified model. So those are the two projects, and I'm happy to answer questions if you have them. Thank you very much.
Hi, I had a question about your FIRE project, and I was wondering about how much of that you think is actively made by the transcription and transcription factors that are being activated, and how much is telling you about, is it organizing the genome
right.
On its own?
Yeah, this is kind of the $64 million question in the field is when we look at three-dimensional architecture, how do we assign causality, right? So is it the case that the 3D architecture is simply a functionless byproduct of the functional activity of things like genes being active, and therefore they construct things by their activity? Using Hi-C, it's not necessarily, you can't directly observe the causality, but one of the reasons to use things like a polymer model is to try to tease that out so that we can ask the question of, what happens if we change things? Does that change the functional process? I think we're a little bit farther away from being able to do that at this point because we haven't included the gene regulation into the model yet, but that would be the ultimate goal. Okay. Thank you very much.